So how did, David and yourself got together, and where did you see that this potential to revolutionize Legionella detection, David?
Well, really comes out of frustration. I've always said we're dealing with 2-week-old data. Literally, the places are shutting down or spending, hundreds of thousands of dollars in filters and just trying to figure out how to exist until they get results back.
For us, for our laboratory, our mission is to provide accurate, fast, and reliable results.
Really what they need to know is that you really don't have to change your procedures on how you're doing, even how you're interpreting samples and how you're collecting the samples and how you're getting the samples to the lab. None of that has to change.
Without my team, I will never be able to reach this level, And my team is not only Diamidex, but it's also all our supporters around the world. And David and Robert are part of the team. So thank you.
Good afternoon, ladies and gentlemen. Welcome back to another very special episode with HC3. I'm joined here with my colleague, Dr. David Krause, and we have got some wonderful guests that we're going to be bringing in. We're going to be talking about the MICA Revolution. That's what I've kind of called it. There's this revolution going on. And then just to bring you up to scratch, ladies and gentlemen, the 1976, there was an outbreak in Philadelphia at the Bellevue Hotel, and many servicemen got ill, and consequently, there was a disease. There was a bacteria that was found on investigation called Legionella pneumophila, and that has since been growing concern ever since 1976. And in fact, part of the problem is detecting Legionella in water systems and also being proactive in a way that you respond quickly. And part of the issue has been that testing has been laborious. It's been slow. We haven't been able to get rapid results, rapid detection of Legionella. But now there is a new revolution. Isn't that right, David? We have a revolution at play, and so we are bringing in the experts today. David, let's welcome our guests. First of all, let me bring on the founder of the MICA Revolution, shall we say. This is Sam Dukan. Sam, welcome. Glad to have you with us. We're all looking forward to chatting to you. And also our next guest is Dr. Florence Wu from AimTech Laboratories. She is with us today. We're going to have a great conversation with her as well. Florence, welcome. And last but not least, the man with the plan. We're bringing him on, Mr. Robert McNeely, who is here to talk to us about training and his French connection as well. Ladies, I would say, gentlemen and lady, welcome. David, it really is a revolution of sorts that's been happening. Why don't you take us through some of that history and what the problems have been before then we jump on and talk to Sam about this revolution?
Absolutely. We have a very esteemed group of researchers and laboratory technicians and consultants here. So, this has really been the triad of trying to fix and figure out Legionnaire's disease. That outbreak that happened in 1976, it wasn't the first time it had happened, but it was certainly the first time it was investigated enough to figure out the cause. Finding it in the people, in the, hundreds of veterans, the American Legion attendees who got sick, that was the easy part. Finding it in the environment, culturing it from environmental samples actually took many years afterwards. And wasn't until almost 1980 before they had, I'll say, more reliable and consistent ways to do this. Unfortunately, what we've done is those initial sampling methodologies and laboratory analysis really didn't advance. since 1980, and we're still using what are fundamentally the same spread plate protocols and multiple plates and almost 2-week turnaround results to get environmental samples analyzed. And so, that has really stymied efforts to effectively respond to outbreaks and also to respond to cases or, issues that arise. You're always dealing with data that's almost 2 weeks old, and the world goes on. So that's been one of the big issues, and that's what we want to talk to you today about is moving from 1980 to 2025.
And that is a massive leap. And actually, I think you're right, David, is that it has been stymied, it's been slow. And when you think about the progression that's been made since 1976 to 2025, literally it has been laborious. But we have a solution now, which we launched here in the US, but has been going internationally for a while, called the MICA Advanced system, which detects Legionella in 48 hours maximum, so 24 to 48 hours. And we have the esteemed professional with us today who invented this, who actually made this breakthrough. Sam, if I can ask you just to go through and tell us a little bit about how did you reach the point of I can actually speed this process up? What happened? What were you doing at the time? Describe to us the whole process.
Okay, so it's a long story, so I will not, even in 1 hour, I will not be able to explain you everything, but just to make it very simple, by training, I'm a microbiologist, and I was head of an academic research group. So, and I was working on aging in microbes. So you see, it's very, very far from Legionella. And to work on this topic, I have asked one of my friends that is a chemist to develop a probe that was able to label specifically the living microbes. And we start with E. coli. We were not starting with Legionella pneumophila. And we discover a new way to label specifically microbes. And we saw that it was possible to do exactly the same, with Legionella pneumophila, with a molecule that will be metabolized specifically by Legionella pneumophila, and that will recover the surface of the cell with some anchor. And by click chemistry, after, we were able to label them. This was done in 2014. So it was just the initial idea, to be able to label specifically Legionella pneumophila with a probe that will be after click and become fluorescent. From this idea, we developed during 10 years a solution. And the solution is the possibility to detect Legionella pneumophila at a stage of microcolony. And combining this probe that labels specifically Legionella pneumophila and this equipment that is able to read, the signal of fluorescence generated by the microcolony, we are able to count them. I make it very short, but it's a long story. And it's not only me, it's a team of 30 people, so.
Brilliant people, a bunch of brilliant people they put together, which Jockey and I talked about yesterday, that they're
we did.
Just brilliant people.
Actually, Sam, he was singing your praises yesterday when we were chatting, and I've never seen Mr. McNeely being so excited about the prospect.
We were lucky because he was in our facility, a few weeks ago, and he saw everything, from the inside, So it's why maybe.
Yes.
Yeah.
Thank you. Yes,
it was like
so.
Charlie in the Chocolate Factory when it was.
I was trying not to get caught up in the chocolate,
So what is fascinating is that you have managed to identify this and really had a breakthrough, because obviously when you test things with ISO, it's not as accurate. Something we talked about as well, Robert and I, yesterday. And this really has had a breakthrough. So how did, David and yourself got together, and where did you see that this was this potential to revolutionize Legionella detection, David?
Well, really comes out of frustration. I've always said we're dealing with 2-week-old data. Literally, the places are shutting down or spending, hundreds of thousands of dollars in filters and just trying to figure out how to exist until they get results back. And when we finally met with Sam and were able to review the published studies, the data, the equipment, I'm a pretty critical person on this kind of material and throw a lot of really hard questions at things. I've seen a lot of things come and go, like my hair. But they have done their homework, and every question I threw at them, how have you validated? What is the positive predictive value, negative predictive value? How has this been used up against existing standard methods? And what are the the potential interferences and ways it can go wrong? And they have been as curious, if not more so, and explored those things up front. And so I felt very comfortable and confident and said, let's give this a try. Let's see how it's working. And, from that point, I said, we have to get this into the hands of a trusted, qualified, competent, great laboratory. And I've known Dr. Florence Wu for, I'll say, a few years. But I
was going to say, what a perfect segue, David, to get this into to bring in Florence.
And I have relied upon her laboratory and their research capabilities and willingness to do the hard work. And I brought it forward, and I think they were willing to be that first adopter, that early adopter, and I think give us a very frank assessment of what they were seeing.
Now, Florence, let me ask you, because now that you've come into this narrative, into this story now, where David has connected with you, and he said, "Hey, look, Florence, I have an idea. I want to bring something to you for you to test." Did you have an early skepticism to this? How did you embrace this in your laboratory?
Well, first of all, I think David's lose more than his hair. He loses memory too. I have known him over almost 20 years. So the respect and the trust is mutual. So when David brought this idea to me, I heard of this matter before. But for us, for our laboratory, our mission is to provide accurate, fast, and reliable results. Since we don't create our own method, so we have to really strict on selecting appropriate method. So with David, I trust David. When he brought this to my attention, we really look at their study. So the method itself is AOAC validated and certified, but we didn't take that as face value. We also did verification study ourselves, and the results are proven. So that's the first step for us. And the fast part is really in line with what we try to do. For over 10 years, we are accredited and PT, certified lab by CDC to do Legionella testing using the ISO method. And our ISO-P for ISO method is like over 20 pages. And we'll need training and repeated training for the technician to really master method. So this, and on top of that is the turnaround time. Most of the customer want really fast turnaround, and sometimes they want the result even before they send samples.
Usual.
I'm not surprised at that. Absolutely.
I know. We totally understand, right? When you're in the field, you want the result fast. So the ISO method is typically 7 to 10 days. We can check the sample by day 4. If there's growth, if it's negative, we have to incubate a longer time. And so this faster turnaround time is really beneficial to our customer, but also have a benefit to the lab, which means that shorter incubation time will allow us to scale without keep adding incubators, right? 2 days, 1 day, the incubation is done instead of waiting for a whole week. So, and then of course the reliability. So reliability for us is both the service and also the result. The result is reliable, accurate, and reliable. In the ISO method, even though it's a standard method, require a lot of training, but sometimes look at the colony, you're just not sure. Like the bacteria, we all know the physiology, the colony, the agar, and whether they're injured or not. So there is always an uncertainty in there when we try to identify Legionella pneumophila. So in our lab, we kind of, to be sure, we actually sometimes need to pick a colony to do DNA sequencing. But this part is usually not requested by customer, so we cannot charge them. So we use our own cost to make sure we generate accurate data. With MICA, we don't need to do the confirmation, and it use AI, machine learning assisted automatic reading. So take away the subjectivity of result reading. I think that's, to me, that's really a good advantage for choosing this analytical method.
I think that's fascinating. I want to jump back into Sam because, as we've heard, Florence has maintained that accuracy is of fundamental importance. And when myself and David and Robert have been chatting, the accuracy of this is like it's out there. It's sky high. It's gold standard. Let's talk a little bit about that accuracy, Sam, and why are you so confident? I mean, we were talking last night when Robert and I were chatting, a 95, 97, 98% accuracy. That is unheard of. How did you get it that specific?
Oh, it's because we have worked several years on the topic. We have tested more than 50 different strains first of Legionella pneumophila, and we train I say my baby, my machine learning. I train it with 50 strain, and after I use more than 2,000 real samples containing Legionella pneumophila inside, and I explain to my baby, look, here is an object that is Legionella pneumophila, and here is an object that is not Legionella pneumophila. So after that, when you train a baby for all these sample, it starts to become a genius. So it's why we have a very good accuracy, And where I'm very proud, it's when you have third-party laboratory that have tested, our solution, they all conclude the same. We are even more accurate than the ISO method. And this especially for the cooling tower where you have a lot of interfering flora. With our system, we are able to count Legionella pneumophila, where the standard method is not able to count because you have a lot of different microbes that cover the plate, So this is also a big advantage of our solution. And we have been evaluated by the Chinese CDC and the European Water Test Network Association and by the AOAC, and the three organisms conclude the same. For cooling tower, even more accurate. Yeah.
Do you know, it's interesting. It's interesting, Sam, because something that Robert and I was talking about, and he maintained this last night, is he'd said, and if anybody's listened to this, obviously you're going to see this, listen to this, and it will be a few days past or whatever. But we were talking about it. Now, you challenged, Rob, let's talk about this, because you challenged consultants out there that were involved in testing to actually test MICA in different laboratories and also ISO. Let's talk about that.
Oh, yes. We have, it was a very important point for us. We send the same sample to 10 different laboratories that are accredited, so, very well-known laboratory. And we send with 1 million Legionella pneumophila per liter. So it's a huge number, and we request the value, and we still obtain result 0. No
That's
Legionella
crazy.
pneumophila, where you have 1 million Legionella pneumophila per liter. So the variability was around 2 orders of magnitude. When we done exactly the same with MICA, with the same sample, the variability was around 20%. So you see the difference, 2 orders of magnitude and 20%. And even some lab obtain 0, We call the lab, we were so shocked, that a lab detect
You
no
didn't believe
Legionella
it.
pneumophila when you have 1 million. And we call them, we say, are you sure? Yes. They say, yes, no Legionella.
That's absolutely crazy, because that's something Robert and I was talking about last night, wasn't it, Rob? I mean, you even set down that challenge to consultants to go out and do that.
Yeah, that's I would say send it around. And this is the thing that I find about the MICA is that you're going to get consistent results. Unlike with ISO, you're going to get consistent results with the MICA. And that's where I think the difference maker is with the MICA compared to ISO, is that from lab to lab you could differ drastically with the ISO. And where with MICA, no matter what machine you use, you're going to get within, a certain variability that would be naturally and that would be expected in results. You will get the same number. And so that's where it didn't matter how well that person was trained, as long as they did the scope, which, Florence could probably speak about, is that it's not, when it comes to a lab, it's not really intensive training and knowledge that you must have to use MICA.
Well, I mean, that's it. And that's another nice segue, because Robert, you went down to Florence's lab when they were putting the test system
Actually,
in.
all of us. Yeah.
All you did. Yeah. And
Yeah.
so
Yeah.
were you shocked, Florence, when this came in and you started to do the testing, that how easy it was to integrate this into your operation?
We appreciate it. I certainly appreciate that. I wouldn't say shocked. It takes a lot to shock me. But I think we appreciate the methods is pretty straightforward, and of course it definitely needs training, right? All the reagents, we need to understand how it works, and technicians learn all the principle behind it and understand where need to take a precaution. But in general, this method is easy to train, easy to learn. And again, the confidence in carry on the procedures and in reading the result, I think that's mean a lot to us.
I think there's a high level of confidence there. I think that makes a difference to all of you guys involved in this, is that maybe before there'd be less confidence in some of the data. There's been times where the data is questionable and you then have to continue testing. But David, how has this changed the industry in terms of trust and confidence in moving forward?
Well, there's two big aspects here. Is let's focus on Legionella pneumophila. There's 62 different species of Legionella that are accounted for at this point, many serotypes. And while, it's been a conservative, and frankly, I think probably a gut feeling from public health agencies to say you need to track and trace and respond to all of these species, Legionella pneumophila has been the cause of 99.9%, all but one of the nearly 1,000 Legionnaire's disease outbreaks in the country. So why are we responding to spending time and resources in dealing with species that in theory can cause disease, but haven't in practice posed a public health risk? So that's been one of the barriers, I think. And people are recognizing that gut feeling is not based in evidence. It's not based in science. It's not based in study or research. The other one is, even if there is a diminution in what you can see, the speed with which you can get final reliable results outweighs any downside. You're able to get results that you can make decisions in and actions in near real time about what we see for any type of chemical test. So I think as people see that and see that they're getting it at comparable cost to existing methods, to which they're waiting for weeks,
Yeah.
that's going to be a huge driver as well.
I think it's also a case, David, of you'll be able to deploy it operationally very quickly to probably expedite remediation process should during an outbreak.
Absolutely. Robert did the math on this. You could, with MICA, if you took your samples on the same day that you took your first samples using the ISO method, you could literally take your samples, get your results, do your remediation, take your post-remediation samples, and know whether that worked or not before you'd ever get your final results using the existing ISO method. So that's a huge benefit, and that literally will save lives. When we extend an outbreak out 30, 90, 120 days, more people are getting sick, and 1 out of 10 of those in a community-wide outbreak are likely to perish.
It's a bold statement, guys. It's a really bold statement to say that this is a lifesaver. But Sam, how do you feel when I know you're proud of your baby, and rightfully so. Well, what you've created is historical in actual fact. But did you feel that this is a lifesaver? This is potentially going to save multitudes of lives all over the world?
It's what we believe, and we push the solution all around the world with the same information and the same explanation. But where I'm very impressed is that in the US, we feel that people are really concerned, by Legionella. In some other country, we explain that, look, with our solution, 2 days it will save life. And labs say, who cares? I'm following the rule. It's enough.
Wow.
Yeah.
It's very strange.
And here we really do recognize that following a rule, meeting compliance with a standard or a rule or a requirement doesn't necessarily manage risk.
Yes.
Absolutely.
We have to be more aggressive than that in order to manage that risk. And it's risk of lives, it's risk of welfare and hospitalization, but it's also a business risk. I can go down a laundry list of hotels that have closed, become bankrupt, never reopened because of Legionnaire's disease outbreaks. And one of those was the Bellevue Stratford. It closed literally a few months after that.
That's right.
It did not open for years.
I think it reopened again in 1998 or 1999, something. No, 1979, something like that. I think it's also important to note that not only dangerous to their business, but even in terms of the investment in their workforce, because when the workforce get ill, they're off. There's increasing costs. There's increasing tax on the organization, and I don't mean tax as in federal tax, but tax in the system, tax in their whole business. How important, David and Rob, anyone you chime in with this, is this in the United States for businesses such as in the leisure industry and the healthcare industry?
Immeasurable, I would say.
Absolutely. I think one of the things that people need to think about is early detection allows you to respond faster and to take measures to remove that contaminant, remove that biological agent before it shuts you down, before it results in illnesses or disease, lawsuits, or just as bad the 6:00 evening news on your front doorstep. I mean, that is a bad Friday afternoon. And
yes, it is.
People, organizations will as people have often asked me when they're the center of an outbreak investigation, is how much is this going to cost me? And I'll say I've never seen a large facility get their way out for less than $250,000 when it comes to remediation, testing, litigation, all of those things. They add up quickly. And if you have to shut things down or bring in bottled water and filters and everything in between, and of course the loss of confidence of your residents, your guests, your patients, your tenants, all of those have a real substantive cost.
Something I was thinking about after Robert and I had a good conversation, and we never talked about this, Robert, and David will understand this, and anybody, Florence here from the States, is we have a lot of natural disasters. Obviously, we've had Hurricane Helene, we've had Hurricane Milton, we've had natural disasters, and I see a potential usage in disaster response recovery because remember just last year when we had Helene, the amount of water, the amount of problem, the amount of pathogens that were found in the water after this. And I don't think this is something that has even been looked at properly in terms of, and maybe this is another thing that MICA can be looking toward, is disaster response. Am I right or am I wrong?
Well, it's going to be more in that secondary phase. So as people are moving back into the properties and beginning to use the infrastructure again and think it's safe to use the showers and the water systems, but nobody told them that they need to hyperchlorinate their buildings' water systems because it's been sitting stagnant for 3 weeks. Yeah. And, the chlorine coming in is much less now than it used to be. That level of understanding really hasn't been a subject of awareness, but we do see a correlation between disasters, storms, and illness in the months after.
I think absolutely right. Definitely the secondary and even an ongoing phase of testing that I would certainly like to see. And how important? So, Robert, down to your side of things. I mean, when people are interested and they want to get this installed, or you're going to be dealing with professionals in the field. Luckily, you went over, and not on a holiday, but you went over on a work trip to France, and you were absolutely excited. You loved it over there because you learned so much. Let's talk about what the professionals have to go through to get this implemented from conception to installation to operation.
Well, I mean, Florence could probably attest to this too. I mean, it's not a huge process, and and this is the thing. Within a month, she's already doing analysis. She's already doing the process, so it's not a huge process. It's just you install the machine, you do the training, you do your validation, and then if you accept it and you think it works, which you're going to. And then you do the validation, the internal validation that Dynadex has required. And that's the great thing about this. And this is for everybody to know. I mean, Florence's crew had to go through this, is that Sam is going to supply you with samples that you're going to have to use the MICA machine to determine those concentrations. Yeah. And you're going to have to be within a certain percentage of that number to be deemed to be able to use the machine. And I don't know any other method, because even if you're elite, it doesn't matter whether you get the good number or not. You just, hey, can you detect it? No, they want to do more than detect it. They want to make sure that you actually can get the number that is actually an accurate number. And so Florence has gone through that, and they have been able to do that, and it's shown so, and so that they've completed that process. And after that, then you're free to use the machine and to market it and supply people with the best type of analysis you can get out today. And that's what Florence and her crew have definitely done.
And they really knocked it out of the park, being able to to, adopt it and become comfortable with it and develop those internal SOPs. And one of those SOPs that, we haven't seen here yet, but I know Sam has seen in other countries, is the need to take those isolates that they grow and actually take it on for serotyping or whole genome sequencing. If and when there's an outbreak, that's still possible, and you can still do that with samples. Exactly. It doesn't kill or injure the samples or the bacteria. They can be brought on for whole genome sequencing and fingerprinting and compared to clinical isolates, which a lot of, the existing, like PCR methodologies simply you aren't able to do that with. So, it really fills a void and a gap that we've had in the United States for some time.
I find it also fascinating, guys, that you can see images. You can actually see a rendition of the images of the Legionella, which is unheard of. I'm not a molecular scientist or anything like that, but for me on the sidelines to look at it and say, wow, I haven't seen any other report that has that. I think that's a very unique selling point or benefit, is to be actually to see an image representation of what you have.
Yeah. Yes.
Because we are microbiologists. We want to see.
Yeah, you want everybody to see what you see.
Yeah.
But you know what?
You know?
Does this help in terms of, this might be for you, David, but seeing the imagery, would this help in terms of litigation on either, defense or prosecution? When you go in to deal with these cases, having the imagery of a potential colony that's colonized your images there, is there a benefit there?
It certainly doesn't hurt. So, human beings in general, we trust what we can see.
Exactly.
Yeah, exactly. Yeah.
Up until now, your reports come back with a name written in Latin and a concentration, and that's all the information you get. And, people can't appreciate what that is. And I've testified in many courts and trials on everything from bacteria to mold to chemical exposures. And I can tell you that the engagement and the believability that I see in the jurors and the judges,
right,
when we have an image that depicts the hazard, the sample, the measurements,
yes,
rather than just somebody telling you a story, you're seeing that story. And I think Diamondex and Sam were brilliant in integrating that into the generated reports, that each sample comes with a image of what it detected, and it's enumerated in that fashion.
Absolutely. And to see examples of that on Florence's website, you can actually see examples of those images as well, which is fascinating. And I kind of want to jump into another subject. I know we're talking about rapid Legionella detection and, getting to the testing, but something that I'm fascinated with as well is that MICA does not just detect Legionella. I mean, essentially, it also detects other pathogens like Pseudomonas, and maybe potentially in the future, I don't know, something that Robert and I was kind of spitballing last night, maybe NTM, so we don't know. Maybe that's something for the future, Sam. What do you think?
Challenge.
Oh.
So I got to poke him, I got to.
It was confidential, so. Okay. So, I mean, right now we have indeed several other microbiological parameters in the equipment. So we are able to enumerate not only Legionella pneumophila with the same approach, but we are able to enumerate Escherichia coli just in 6 hours instead of 24 hours.
That's
crazy.
Which is
very fast. 6 hours, same day, result. We will have total coliform very soon, by end of the year or beginning of next year, we will have total coliform in a very short period of time. Next year, we will have Pseudomonas aeruginosa, indeed. And we receive a lot of feedback on non-tuberculosis Mycobacterium. So we are starting to think of developing a solution. I mean, when I say that, we have already proof of concept, because we have proof of concept for many, many different microbes. But we need to know if there are a need, because we have so many ID, because we are scientists first. So we need to see if there are really a need. So apparently there are a need for non-tuberculosis Mycobacterium. So we are thinking to develop a solution for that based on the proof of concept that we have. Yes.
Now, ladies and gentlemen, if you're listening to this on our podcast and all the podcast networks, you're not going to see Rob McNeely's face just lit up, because we were talking about this last night and he was like, yeah, I can see Sam, going for this and looking at NTMs in the future. This is, a potential new field. And it's great, Sam, that you brought that up. Because then people who want to, bring this system in to their labs or into their operation, I hate to use this little term, but it's going to grow with you, right? Obviously,
the baby keeps on growing.
It's going to keep on growing. How do you feel about that, Florence, in your lab? With the potential that you can grow and this can move into new detection.
We're excited. We're excited to grow with the technology, so it makes us to be more effective, more efficient, to reach out to a broader customer base. Any technology advancement, we'll be able to use. That whatever Sam can provide, we'll be able to use. We see ourselves as the bridge between the technology and our customer and the data. So any technology can enable us to provide, again, accurate, fast, and reliable results. It gives us opportunity to grow, give us opportunity to contribute more to the society.
I love how we talk about growing into the future, because I feel, from what I've learned about Sam and the organization, from the nuts to the bolts to everybody that's involved, that Rob and I were talking about, Sam, you're a very futuristic company. You've already, harnessed the power of AI. Are you going to continue to keep harnessing that as you move into new fields? Are you forward thinking all the time, trying to make it better with AI?
So we are thinking for the new solution to develop new approach of machine learning, but as soon as a solution is developed and installed, in many different customers, we cannot modify the machine learning that is inside, because we have already laboratories that are ISO 17025, so they have been certified for the solution. So we cannot change the machine learning that is inside. And what is very important, because everybody, since few years are talking about machine learning, artificial intelligence.
Yeah.
What is very important to understand is that the machine learning is done in our laboratory, not in the equipment. In the equipment, there are no evolution of the equipment. It's fixed. So there are no more machine learning inside the machine that Florence have, of all our customers have. So it will not evolve during time. So the baby is frozen and it will not change, So this is very important, because you cannot be certified ISO 17025 if your algorithm is evolving every time,
Yeah. And we certainly don't need any, and we don't need any equipment, any AI hallucinations.
Exactly.
On reading these. So
I was going to say, you still need human intelligence involved in this. You still need the human ability to discern and to look at that data and to develop it. And at the end of the day, even though that you have AI in a certain way, Sam, that you're utilizing it, isn't it really the human input that's putting all that data in and training the AI? So at the end of the day, it is actually being human developed.
Exactly, because we have some expert in the lab, that zoom to all the image and check first by eye and explain to the machine, here is a positive. So without the human, the machine will never be able to do it. So the machine needs to be trained by the human first in order to be able to detect after. So it's totally different, you know. And one point that have not been, pushed out is with one equipment, you could perform until 500 analyses per 8 hours. So it's a really high throughput system. So
Yeah.
for laboratory, you know, that want to grow with us and increase the business, you know, they will be able to receive a lot of sample with no need of having many equipment, you know. Just one equipment will be enough for almost all the laboratory.
And it's the same piece of equipment they're using
For
for
all.
Legionella that's also reading samples that would be analyzed for Pseudomonas or E.
Yeah.
coli. And so you really do have a kind of, I'll say, a French
Platform.
Army knife instead of a Swiss Army knife, but you have a multi-tool that can do many jobs very well.
That is actually is a fantastic analogy, David, to say that a French army knife. That was very, very very good of you to move to that one. But it's true. It is like your Leatherman. It has so many tools that it can utilize, and again, will continue to grow. Robert, what do you feel that I would say professionals out there need to know about this? Because we're talking about the labs and how important it is, but what about the consultants? And we've got a great conversation coming up with some consultants as well. But what do they need to know, Rob?
Really, what they need to know is that you really don't have to change your procedures on how you're doing, you know, even how you're interpreting samples and how you're collecting the samples and how you're getting the samples to the lab. None of that has to change. You can still continue to do that. It's just you're going to get your results significantly faster. And you can really count on those results. That's what they need to know is that I know from lab to lab that they're different. Even within the same lab, it could be just who's analyzing it could change on the results. I guarantee you send the samples to Florence at AIMTech, you're going to get an accurate result, period. And that's what they need to know is that you can be confident that this machine is, and the process, the method, the total method, the solution, as Sam likes to call it, will be correct, accurate, and precise.
Yeah, and not to speak ill against the existing method, but the existing ISO method and the elite certification is really built upon a fairly weak expectation of reliability. There was a very interesting paper published back in 2011 by some of the members from the CDC that did a pilot study on the elite certification on accuracy and precision of Legionella. And you know, I'd love us for us to leave a link to that in the
we can do that. Yep.
In the box here. But you know, when you read that and you realize how imprecise and I think they showed that really the existing methods that were being used by elite certified labs consistently underestimated the actual concentrations of Legionella in those samples by 1.25 logs. It was not just error, it was bias. It was biased low by, over an order of magnitude. And then, the inability, while they have detection limits that are 10 CFU or, 1 or less than 1 CFU per mil, when, 90% couldn't detect it when it was less than 10% or 10 CFU per mil, that really is telling. Yet that is the standard that we're comparing it to.
Yeah.
So this is just head and shoulders in its repeatability and reliability, and I think will allow us to have greater confidence in the results and protect public health.
David, let me ask you, do you feel that, and it's something Rob and I had just skipped over last night as well, that there'd be some kind of, maybe not legislation is perhaps the wrong word to use, but some kind of derivative, you have to use this system, insurance companies or the make it that in order, because it's so accurate, because it is a breakthrough, that it will become standard, that MICA Advanced becomes the standard in the industry, you have to use that. And that maybe
doubt
insurance
we're gonna
companies will say that.
Yeah, I doubt we're going to see legislation. It could happen, but often what happens in these cases is you get performance criteria, and the market drives you to using the most accurate, reliable, precise, and and timely results. So often what will happen is they'll say, these are the performance criteria you have to meet, or your laboratory needs to meet, and faster, better, and at the same cost, or more reliable. Those kind of things, I have every bit of confidence that that would probably be the most likely route in which it goes. But we do have legislation. We do have legislation in New York City, New York State, and implementing currently in New Jersey. So that's becoming more common and more frequent. Hopefully, they are picking and going to rely upon reliable measurement methods.
And I think since we've launched it in the US and made a big issue about it, that there has been, the jungle drums have been beaten. We have been hearing them. You have been in conversations, and it seems to be that there is less pushback and more curiosity about this revolution, if you like, this fast Legionella detection, the ability and the potential it has to revolutionize the industry.
Absolutely. People are very curious and want to move forward with this. Having conversations every day with private end users, whether they're water treatment companies, consultants, public health officials, state labs. So, I mean, we're moving forward with purpose.
Let me ask this question. I know the answer to this question, but I've got to ask it because people will say that we don't want to, as you mentioned, bias. Bias is the bane of scientific inquiry. And so is there any, Florence, Sam, is there any downside to this at all?
For us, the downside is we probably have to maintain a good relationship with our supplier.
Good answer, Florence. Good answer. I like it.
Chocolate chip cookies.
Yeah.
Oh dear, that's good.
That's good.
So the upside, I think the upside outweighs downside a lot. We talk about accuracy. In analytical service, the accuracy is actually directly related to less handling. Like in ISO method, there's a lot of handling. There's a memory filtration, and there's suspend the filter, recover the bacteria from the filter itself. The filter recovery itself is a science. Depends on what the filter you use, how long you vortex it. So the less handling, less step in analytical analysis can result, less error. So that's where I think the bias come from. It's from the method itself, not from the training or so the method itself gave lab the confidence that our data is less impacted by human error.
Yeah, I think that's very important. Actually, yeah, exactly. Couldn't say it any better. Is less human error, because at the basis of it, it comes down to human error most times.
And it gave us I as a lab, one of my job is educate our customer. Education meaning I will give them a choice of method and explain the advantage, disadvantage is method. Yeah. So in the past, we have this ISO, and then we have a qPCR. So my explanation to them, if you want a viable result, then you have to wait 7 days or 10 days. If you want a fast result, then you don't necessarily get the viable count with a qPCR. So either way is not ideal. So in comparison, MICA is ideal in terms of both fast and viable result.
That's absolutely. It must be great for you to hear this, Sam. I can see you smiling. It is your passion. I know.
Yeah.
Ladies and gentlemen, I can't say ladies, but ladies, and I want to thank you all for joining us this afternoon, and hopefully we'll have you back. And anybody who's got any questions, we'll send them in to us. if you have got any questions, if you're listening out there or you're watching this and you've got any questions for Rob or David or Florence at AIMTech, then please send them into us. It's hc3fl.com. Sam, you have absolutely revolutionized this industry. And from someone like me who's a beginner, you know, I'm not a microbiologist in any way, but I find this fascinating because I'm curious. And since working with David and Rob, I'm even terrified. I look at my shower heads and God knows what else, and you know, I find myself, you know, negating going away from fountains and hoses, and God knows they've terrified the living daylights out of me of it. And as a Scotsman, I've learned to say Legionella pneumophila instead of Legionella pneumophilia, which is what I would say in Scottish. Guys, thank you so much, Florence. Fantastic. Thank you for coming on and sharing your experience with this. Wish you all the very best with it. And Sam, we're going to have you back on again. Let's have final thoughts. Sam, give us your final thought, then we'll move around the room and we'll finish off there.
So what I will say is that it's not me, you know, it's the team. It's, you know, without my team, I will never be able to reach this level, you know. And my team is not only Diamidex, but it's also all our supporters around the world. And David and Robert are part of the team. You know, so thank you.
Wow. That's fantastic. Florence, over to you.
Oh, thank you for this opportunity, and thank you for bringing the technology to us so we can do our job better and meet the client's needs better.
Yes, and if anybody's listening out there and you need some Legionella testing done fast detection, then get in touch with AIMTech. We'll have a link below this conversation to AIMTech and everything else as well. David, final thoughts from you?
Well, I'm just glad to be a part of this team here and be able to bring forward what I see as just a fantastic and revolutionary approach to a growing problem that you know, we really do need to step up and make some changes so that we can stop the number of cases, outbreaks, and deaths that happen from this every year. So again, thank you for everyone being part of this, and I look forward to the years ahead.
Fantastic. Robert, final words, my friend. What have you got to say?
Well, I think it pretty much covered everything, but I would have to say, when it comes to Diamidex, I think Sam is correct. He's the pinnacle of the brilliance there. But man, what his brilliance was surrounding himself with absolutely brilliant people. I met all of them, and they are all just amazingly brilliant and and passionate about what they're doing. And that's why, you know, when me and David were thinking about, well, what lab could we really bring this to? And we were thinking, okay, who do we think is the pinnacle of labs and expertise on being able to really evaluate something?
Yeah.
Florence Wu. I mean, that was it. And that's why we went to her, and we were so happy that she accepted, because we were kind of, well, let's see, you know, and thank goodness that, you know, she trusts David, because I don't think anybody else could have got her to do it other than David. And so, and I think with that, she's seeing what this is really going to do. And I think people are going to use this, and they're going to see the real benefit of it. It's going to take a little bit of time, just because, you know, people are a little apprehensive about something that's this revolutionary, this amazing, because, you know, everybody's going to be like, what's wrong with it? Come on, there's got to be something wrong with it. This is too good.
I remember.
I remember. Yeah, you know.
Yeah, there's got to be a skeptical side.
Yeah. So and good luck, you know. I think Sam's done a real good job of that, is saying, prove me wrong. Prove me wrong. And nobody's been able to.
And I think with the accuracy and the quality of the system, you know, I know it's, maybe Florence don't want to hear this, but other labs have got to get on board as well. And so, you know, I think Florence is leading the way in that side of things. But, you know, there's other labs that are going to be left behind. And I'm so glad that Florence has joined the team and has got this up and running. So, guys, thank you for being with me again. I look forward to chatting you. Ladies and gentlemen, you have witnessed history in the making. It says history repeats itself. From 1976 to the outbreak, everything has been slow. There's never been a breakthrough until now. And now you're actually seeing MICA advance as that breakthrough. It's cutting down the time. It is saving money. And more importantly, it's saving lives. And that's what we're all about. So please share this with people. Let them know. If you're in the industry, let them know about this massive breakthrough that could save lives. It is so important. Join us again for other discussions. You'll find Dave and I will be up very soon with some other people out in the field where we'll be taking their questions and talking to them. Contact us at hc3fl.com and also Diamodex. You can get them online. We'll have links to everybody below this. Sam, Florence, David, Robert, thank you for joining me today.
Thank you.
Guys, this is Emerging Pathogens, brought to you by HC3. Keep safe, and we'll see you in the next one. God bless.